Hey everyone. Uh, Thanks for having me today. Really excited to talk about this. So my name is Michael Silverstein, and I'm from Boston University in Daniel Scray and Jennifer Bhatnagar's lab. And today I'm excited to tell you all about a recent study I did that investigates this relationship between metabolic complexity in environments and how that drives the divergence of microbial communities. Um, but before I get there, cool. Before I get there, I actually want to talk a little bit about the motivation for my research, which has to do with using microbial communities to mitigate climate change. So as I'm sure everyone here knows, greenhouse gases are increasing the rate of climate change. And whoa, oh, maybe I'm going backwards. Here we go. Okay. But as some of you may not know, at least I didn't know until I started doing this research, the carbon from microbial necromass can stabilize with soil minerals for hundreds of thousands of years. So this is really, really cool. And when I learned about this, I started wondering, is it possible that we could modify or engineer existing environmental microbial communities to actually increase their rate of carbon stabilization, thereby mitigating climate change? Now, there's a lot of things that go into this question, but one of the key ones that I was interested in recently is, in order for this type of engineering to happen, it has to be the case that the microbial community in one environment the environment has to be able to host multiple microbial communities, because it has to be able to host its current one. But if we want to change its function, then it must be the case that it can host other microbial communities that have different functions. So let me illustrate a little bit what I mean with this schematic here. So in this cartoon over here on the left, we can think of this x-axis as like our PC1 in species space. So as we move around on this x-axis, imagine that we're going through different microbial communities. And on the y-axis, imagine that we have some function of the community that we're interested in. In this case, maybe it's carbon stabilization. And for different species compositions, we're going to have different amounts of this activity. So in this case here, where this might be in an environment where there's only one stable state, it might be really hard to do something like microbiome engineering, because if we tried to perturb this community, we're just going to go back to that single stable state. We're not going to be able to have any other level of activity. Can't do microbiome engineering. However, if an environment supports communities in this way on the right, where there's multiple stable states, then maybe it's the case that we can actually shift between these stable states, find the one with the maximum level of activity for whatever we're interested in. Ideally, this super high peak in the red there. So this thought experiment led me to this other question, which was, what determines the number of microbial communities or states that an environment can host? And when I thought of this question, I was reminded of some previous studies, which I'll show the results of now. So this is a study from 2018, where the researchers went out to a bunch of different soils, collected communities from different soils, brought these different communities back to the lab, and grew them all in a really simple environment, just on glucose minimal media. And what this plot is showing here, each of these colored lines is the trajectory of a different microbial community over time. The distances between the points represent how different the microbial communities are in their taxonomic composition. And initially, those circles on the outside are showing the initial states of these communities, where we see initially each of these communities start off really different from each other taxonomically, but over time, they become a lot more similar to each other. This is sort of like the first example I showed of a single state. However, this is what I'm showing on the right here is a different experiment, very similar setup, except they actually sourced microbial communities from pitcher plants, brought them back to the lab, and grew those communities in acidified cricket media, far more complex media than just glucose. And there's a really different story here. We see that these the squares that I'm showing here are the final states of communities after 30 days or so. And we see that there's a lot of divergence. So the average distance between all of those final states is really far from each other. In fact, it almost looks like these points are kind of converging to different stable states in this space. So when I was thinking about these two different experiments, this led me to generate this hypothesis that maybe it's the case that complex environments lead to more divergence in taxonomy. I'm just basing this off of these two studies to test this rigorously. I wanted to do my own study. So I went out into the woods around here, collected some soil, 6 different communities, brought those back to the lab, and to sort of test this experiment, grew these communities in conditions of increasing metabolic complexity. So going from citrate, glucose, cellobiose, cellulose to lignin, as we move that way, we're looking at compounds that are more complex, they're bigger, They produce more byproducts as they're metabolized towards central carbon metabolism. So I grew these in single metabolite conditions, and I also grew them in combinations of each other. Did this experiment for about 30 days, passaged every 3 days, and then performed 16S sequencing. So what I'm going to show you now is the primary result from the study. So just to start off, what I'm showing, if you look at this inset in the top left, this is the trajectories of my communities that grew in the citrate condition. So once again, the circles at the perimeter, those indicate the initial communities. So we start off with communities that are quite different from each other. And then over time, they like completely converge to each other. So in the end, they kind of had the same composition. And we can measure, that's qualitative, we can measure the pairwise distances between them to get some quantitative amount of this divergence. And now as we move from the left to the right, as the colors go from light to dark, the conditions are getting more complex. So if we just look at these blue boxes for a moment, we see that as the conditions get more complex, we get more divergence. And we can return to this qualitative depiction up there for my lignin condition, where communities start just as different from each other, but here you see that they kind of resolve to, it almost looks like they're clustering to two different states. So we get more divergence when there's more complexity. And this story actually repeats itself in our mixed metabolite conditions. So in orange, we just have these mixtures where the lightest orange there is citrate and glucose. The darkest orange is when we include all of our metabolites. And we call this the divergence complexity effect to describe how in increasingly metabolically complex conditions, our communities diverge more from each other. So once we saw this result, we were interested in trying to understand what is it that drives this effect. And there's a couple of ways we went about this, but I wanted to share one of them with you, which was to use these consumer resource models, which are these computational dynamic ecological models. So I'm just going to explain this consumer resource model from very abstract level. So there's two components to consumer resource model. There's your consumers and your resources. So consumers are defined by what resources they prefer to consume. So in this case, we have these two bacteria here are your consumers. The blue bacteria in this case prefer blue resources. The orange ones prefer orange resources. So that's the consumers. The resources are these circles, and we can define resources in these models based on what resource does it transform into after being consumed. So in the dashed box over here, we see that this dark blue resource is transformed into this orange resource after consumption that is then made available to the rest of the environment. So with these two things like cross-feeding and competition, are all that just emerges for free, which is really nice. And how I'm depicting this concept here is I'm sort of trying to represent the trophic structure of metabolism that exists in nature, where we have a hierarchical consumption of complex to simpler metabolites. So, in the natural world, we have something like a leaf falls on the ground. There's tons of complex stuff in there, and then there's microbes that specialize. They have the enzymes to break down that really complex stuff that makes simpler stuff. Then the next group specializes in that, and so on. But because we're using a computational model, we can create these relationships in any way we want. So one way that I modeled my experiment was to use communities, to use a world where we have this trophic structure with this hierarchical degradation of resources with specialists at each trophic level. But we could also create a model with totally random structure, where we have consumers that consume any resources and resources that transform into any other ones. And one reason why we might actually want to do that is that if we create models with a combination of all of, with trophic resources, trophic consumers, random resource transformations, and random preferences, and we do, all four of those combinations, it would allow us to probe what is the role of consumer preferences or resource transformations in this divergence complexity effect. So this gives us a toy box to kind of test how the structure of metabolism itself might play into the effect that I observed. So I'm about to show you some results from our computational simulations, and I'm going to show them in this grid here. So the result that I'm showing now is the result from the simulations where we have random resource transformations. So here's a world where resources just transform into random other ones. And also in this world, consumers prefer random things. And in this world, kind of unsurprisingly, I think, we see that there's no difference in divergence between our different conditions. I mean, our different conditions are kind of meaningless in this case because resources are just transforming randomly. And now what I'm showing are the results from the other parameterizations of our model, and something very interesting came out of this, and I'm highlighting that in red. So the results in red in A and B, we see that we actually reproduce our divergence complexity effect. So as the conditions get more complex, we see that they diverge more. And what's interesting is we only see this in that first row there. In this first row, this is where we have trophic structure and resource transformations. So this is the case where complex resources break down into simpler ones subsequently. And in this case, we reproduce our effect, even when consumer preferences are random, which is the case in B. So even when consumers eat anything, as long as there's a hierarchical structure of the resources, we still reproduce our effect. And also interestingly, without structure in our resource transformations, we totally lose our effect. And just as a reminder, this is the result that we had from our experiment. So I don't have time to discuss the other things that we did, but this is just to show on the top here, these are some other experimental results. And on the bottom are the corresponding results that we were able to reproduce with our simulations, and they match quite well. So in conclusion, we identified this thing that we like to call the divergence complexity effect, where divergence correlates with metabolic complexity. We think that based on our simulations that this could emerge from the structure of metabolism itself in some way, specifically in the hierarchical transformation of resources that happens in nature. And for me, one of the exciting outcomes of this is that I think that this, provides some hope that it could be the case that complex environments like forest soils might be susceptible to microbiome engineering since they can host different types of communities. Just like to thank everyone from my labs. For people who are interested, we have this paper up on bioRxiv and it's under review right now. So happy to take any questions.